Can your gut bacteria change how you respond to antidepressants? Depression or major depressive disorder is one of the most common mental illnesses in the world. It doesn't just cause sadness, it can drain your energy, affect sleep, change appetite, and make even small tasks feel overwhelming. For many people, it can become so severe that it interferes with work, school, and relationships. SSRIs, or selective serotonin reuptake inhibitors, are commonly used to treat depression. Although they are commonly prescribed, about 30% of patients don't respond well to these drugs. Scientists discovered that trillions of bacteria living in our gut, or the gut microbiota, may play a role in this resistance. The gut bacteria doesn't just help with digestion, but they can also influence our brain and mood through what's called the gut-brain axis. They produce molecules that act like chemical messengers, affect stress hormones, and even interact with nerves that connect to the gut and brain. Recent research has found that people who respond well to SSRIs have different gut bacteria and metabolic patterns compared to those who don't. This study looks at how the gut microbiota might influence whether antidepressants actually work. It suggests that certain gut bacteria and the chemicals they produce could help predict or even improve how people respond to SSRI treatment.
After 6 weeks of SSRI treatment, researchers compared the gut microbiomes of patients who responded to the medication with those who didn't. They found clear differences between the two groups. People who responded to antidepressants had higher levels of beneficial bacteria, such as bifidobacterium and lactobacillus, which are well known for supporting gut health, reducing inflammation, and even influencing serotonin production in the brain. In contrast, people who did not respond had more bacteria associated with inflammation and stress, like allostipes and Prevotella. These microbes have been linked to higher gut permeability and increased inflammatory signaling, which both can negatively impact mental health. The types of microbes present seem to shape how the body processed and responded to antidepressants. When the researchers looked at what these microbes were actually doing, they found another key difference. Responders had gut bacteria that were more active in producing beneficial metabolites, especially short-chain fatty acids like butyrate and propionate, which are well known to support the gut lining and regulate immune function. They also had higher levels of molecules tied to neurotransmitter activity, including tryptophan and GABA-related metabolites, which help reduce serotonin and calm neural activity.
A total of 126 patients with major depressive disorder from the affiliated mental health center of Jiangnan University were selected for only having a monotreatment of SSRIs rather than using multiple drugs. The researchers controlled for other comorbidities, antibiotic, probiotic, prebiotic supplementation, and those at risk of immediate harm. They were then divided into responder and non-responder groups based on how they reacted to one of 3 SSRIs, including citalopram, paroxetine, or fluoxetine. These all started at a dose of 20 milligrams and were titrated up to 40 milligrams depending on how the patient responded. While the researchers allowed the use of benzodiazepines in the short term when necessary, they controlled for polypharmacy by restricting the use of other antidepressants, antipsychotics, and mood stabilizers. Before and after SSRI treatment, the participants of the study were evaluated using the Hamilton Depression Rating Scale. This widely used tool enabled the researchers to decide which individuals would be grouped into the responsive and non-responsive groups of their study based on the patient's relative rate of symptom reduction following SSRI treatment. But how did the researchers actually evaluate the level of gut microbiota within their human patients? Well, the answer is they used fecal samples. Patients provided fecal samples from which DNA was extracted and used to perform PCR in order to sequence part of the 16S rRNA gene. Meanwhile, the metabolites in the samples were analyzed via liquid chromatography, mass spectrometry.
So what do the study's findings tell us, and where do we go from here? In summary, this study revealed how the gut microbiota may influence how patients respond to SSRI treatment for major depressive disorder. By combining microbiome sequencing and metabolomics, the researchers uncovered specific microbial and metabolic patterns that distinguish patients who respond well to antidepressants from those who don't. They found significant differences in both the diversity and function of gut microbiota between the two groups, and certain bacterial species and their related metabolic pathways, particularly those tied to neurotransmitter production and energy metabolism, were closely linked to SSRI effectiveness. To take it a step further, The researchers built machine learning-based models that accurately predicted treatment response based on a patient's gut profile. This validated the idea that understanding a patient's microbial composition and metabolic signatures could serve as a powerful tool for predicting who will respond well to SSRIs. These findings open new directions for personalized medicine in treating depression. By understanding a patient's unique gut microbiota, clinicians could tailor treatments to improve antidepressant response, moving beyond the traditional trial and error approach. Looking ahead, future research could test dietary modification and probiotic supplementation as strategies to adjust the gut microbiota and enhance SSRI effectiveness. Meanwhile, the same machine learning models developed in this study could evolve into clinical decision-making tools, helping doctors design personal treatment plans that optimize outcomes and improve long-term prognosis. In the end, this research builds a foundation for microbiome-guided antidepressant therapy, a future where understanding your gut may help personalize your plan to better mental health.